Hi, this is Pratima and you are watching Planet Physiology. Today we shall discuss some important MCQs related to nerve physiology. Actually, this is the part which students don't pay much attention to. So I created a separate session dealing with only the MCQs on nerve physiology. Okay, coming to the first question. Development of myelin sheath in peripheral nervous system depends on A. Oligodendrocytes, B. astrocytes, C. microglia, and D. Schwann cells. Yes, a simple MCQ. Correct answer is Schwann cells. In CNS, myelin sheath is led by oligodendrocytes, and single oligodendrocyte can lay myelin sheath around many axons at a time, while a single Schwann cell lays myelin around only single axon. Next question. Large myelinated nerve fibers differ from unmyelinated fibers in that former are characterized by A. Solitary conduction, B. Axons are buried in the Schwann cells without wrapping of myelin, C. Conduction is a continuous process, and D. Examples include all postganglionic fibers of autonomic nervous system. Yes, another simple question. Myelinated fibers show solitary conduction. That is, action potential jumps from one node of Ranvier to the another node, and hence the conduction is faster in myelinated nerve fibers. Next question. Not true about myelinogenesis. Option A. Process of deposition of myelin sheath around the axon. B. Begins 1 year after the birth. C. In peripheral nerves, Schwann cells are responsible to carry out this function. And D. Oligodendroclia are responsible for it within CNS. So now here question is not true. So option A, C and D all are correct. So not true. The wrong statement is myelinogenesis begins 1 year after birth. Process starts during fetal life itself. Myelinogenesis is a process of deposition of myelin around axon. Coming to the next MCQ. Which part of the neuron has the highest concentration of sodium channels? Options are A. Dendrites, B. Cell body, C. Initial segment, and D. Synaptic knob. When this question was asked, to our surprise, almost 50% of the students opted for the option D, synaptic knob. So what do you think? Okay, the correct answer over here is initial segment. Actually, this synaptic knob is the terminal part of the axon that participates in formation of synapse. It has more number of voltage-gated calcium channels which help in release of neurotransmitters. If you see the different parts of the neuron, there are just 20 to 75 sodium channels per square micrometer at the axon terminals. In the cell body or the soma, the number is 50 to 70. Then about 350 to 500 in the initial segment, while 2000 to 12,000 at the nodes of Ranvier, and less than 25 on the surface of myelin sheath. Now, since nodes of Ranvier is not mentioned in the options, the correct answer over here is initial segment, which is located just next to the axon hillock, as you can note in this picture. This region is the site for generation of action potential in case of motor neurons, while in case of sensory neurons, action potential is generated at the first node of Ranvier. Please remember this fact also, because MCQ can be asked on this particular information as well. Next question. Proteins associated with synaptic transmitters are synthesized in. Here are the options. A. Nerve fibers, B. Endoplasmic reticulum of the cell body, C. Dendrites, and D. Synaptic vesicles. Since the question deals with neuropeptides, as usual, proteins are always synthesized in endoplasmic reticulum, which are then processed and packed by Golgi apparatus. So these neurotransmitter vesicles are then transported from soma to the axon terminal by fast anterograde axonal transport. Hence, the option B is correct answer. If you see the students' response to this question, about 1/3 of the students have opted for option D. So let me clarify that. Small molecule neurotransmitters like acetylcholine, dopamine, GABA, catecholamines are usually synthesized in synaptic terminal and then are transported into the vesicles for storage. These are not proteins. The only neurotransmitter synthesized in the synaptic vesicle is norepinephrine. So since question here is associated with proteins with the synaptic transmitter, the correct answer is endoplasmic reticulum. Okay, the next question. What does indicate that a nerve has been excited? A. Production of generator potential. B. Production of an electrotonic potential. C. Production of catalectronic potential. And D. Production of wave of depolarization. So whenever we say that the nerve is stimulated or excited, it means action potential is generated in it. So the generation potentials, electrotonic potentials, catalectronic potentials, all are different types of generator potentials and not the action potentials. Wave of depolarization is nothing but development of action potential and hence option D is the correct answer. Next MCQ. Not true of refractory period in a nerve. Now here question is not true. So the statement which is incorrect regarding the refractory period should be the answer. Option A. It is the length of the time it remains non-responsive to re-stimulation. B. Increases with the diameter of nerve fiber. C. It puts a limit to the frequency at which nerve can conduct impulses. And D. Because of it, a nerve can conduct maximum of 1000 impulses per second. This is how my students have chosen the answers. What do you think? What is your answer? Okay, let us see. Option A is correct meaning of refractory period. And unless the nerve comes out of the refractory period, it cannot generate the next impulse and hence it puts the limit for frequency of action potential. Hence, option C and D are also correct statements with respect to refractory period. So automatically the answer for this question is option B, increases with the diameter. If you observe this table, as the diameter of the nerve fiber increases, the spike duration and hence the refractory period decreases. Absolute refractory period for type A fibers is about 0.4 to 1 milliseconds. Type B fibers is 1.2 milliseconds and type C fibers is 2 milliseconds. And we know type A fibers are large diameter, B are medium size and C are the small size diameter fibers. Next question. Material responsible for maintaining integrity of the axon is A. RNA, B. DNA, C. Neurotrophins and D. Lipoprotein complexes. Okay, the correct answer is C. Neurotrophins. Neurotrophins are the proteins necessary for survival and growth of neurons. Some neurotrophins are produced by the muscles or the other structures that the neurons innervate. Other neurotrophins are produced by astrocytes. These proteins bind to the receptors at the nerve endings and then are internalized and then transported by retrograde transport to the soma. Here, they initiate production of proteins necessary for development, growth and survival of the neurons. Some neurotrophins are produced in the neurons and are transported to the nerve endings. They maintain integrity of the postsynaptic neurons. The first neurotrophin that was characterized was nerve growth factor. Coming to the next question. Chromatolysis is seen in A. Demyelination, B. Multiple sclerosis, C. Section of axon and D. During nerve activity. Chromatolysis is disappearance of nasal granules and the correct answer here is option C, section of the axon. Because we are talking about the sectioning of the nerve fibers, Just read about the Wallerian degeneration also. It is nothing but the degenerative changes taking place in the axons distal to the site of injury. Because MCQ can be constructed on this part also. And if you see this poll, this correct answer was given just by about 10% of the students. Most of the students have opted either option A or B. So actually, multiple sclerosis is autoimmune demyelinating disorder which damages myelin sheath and hence the conduction velocity in the nerve fibers. So option A and B are same. Multiple sclerosis is associated with demyelination. In contrast, whenever there is sectioning of the axon, soma of the injured axon shows chromatolysis where nasal granules disappear. Next question again related to multiple sclerosis. In multiple sclerosis, statements are A. There is excessive deposition of myelin around the axons. B. Thickness of the myelin increases. C. There is patchy destruction of myelin. And D. It is associated with increased conduction in the axons. As we have seen just now, multiple sclerosis is autoimmune disorder associated with destruction of the myelin and hence decrease in the conduction velocity. Therefore, option C is the correct answer. In multiple sclerosis, there is patchy destruction of myelin. Coming to the next question. This question is from MGR University, February 21 paper. The transport of proteins and polypeptides synthesized in soma of the nerve cell to the axonal ending is called as A. Retrograde transport, B. Exoplasmic flow, C. Transcytosis, and D. Exocytosis. Yes, a simple question. Correct answer is B. Exoplasmic flow. Exoplasmic flow can be anterograde, that is from soma to the axon terminal, or retrograde, that is from axon terminal to the soma. But here the question is stating proteins synthesized in soma are being transported to axonal ending, so the answer is exoplasmic flow. Next question. Propagation of an impulse in one direction along axon to its termination is called A. Antidromic conduction, B. Orthodromic conduction, C. Anterograde conduction, and D. Saltatory conduction. Okay, here the correct answer is option B, orthodromic conduction. Now, even though both the terms orthodromic as well as anterograde are used for forward conduction, with respect to impulse, the term orthograde conduction is commonly used, while anterograde is mainly used for axonal transport. And all of you know saltatory conduction is jumping action potential from one node to the next node, which can occur in both the direction. Okay, the next question is, true about nerve impulse is A. Travels in one direction at synapse, B. Travels in one direction along the axon, C. Travels with the speed of electric current, and D. If the current is increased too slowly, the nerve responds very fast. Yes, correct answer is option A. It travels in one direction at synapse. If the axon is stimulated in the middle, impulse can travel in both the directions. But the antidromic or retrograde impulses die out at the first synapse itself. Velocity of impulse conduction is much slower than that of electric current and ranges between 0.5 to 120 meters per second depending upon the diameter and myelination status of the fiber. And if current is increased very slowly, nerve fibers do not generate impulses, and this property is known as accommodation. So the correct answer is option A. It travels in one direction at synapse. Here is another MCQ which almost 2/3 of the students couldn't answer correctly. The presence of calcium on nerve membrane may play a significant role in which of the following processes? A. Operation of sodium pump, B. Regulation of potassium outflow, C. Keeping sodium gates closed, and D. Preventing protein anions from going out. So you can see the responses from the students. Okay, the correct answer is C. Calcium ions help in keeping voltage-gated sodium channels in closed state, and thus they prevent unnecessary activation of nerve fibers. In case of hypocalcemia, sufficient calcium ions are not available to keep sodium channels in resting state, and hence the nerve fibers become more excitable, leading to excessive muscle contraction, which is referred as hypocalcemic tetany. So this is the basis behind hypocalcemic tetany. Next question. Conduction in which type of the nerve fibers is blocked maximally by pressure? Option A, C. Option B, A. Option C, B. And option D, sympathetic nerves. This is the factual knowledge with applied importance. So here what you are supposed to find out? Which type of nerve fibers are maximally susceptible or sensitive to pressure? And yes, those who know Erlanger-Gasser classification, they can easily answer this question. It is type A fibers. This forms the basis for Saturday night or Sunday morning paralysis as well. Because of association of deep sleep with alcoholic intoxication, the syndrome is most common on weekends, where if the individuals sleep with their arms under their heads for long period, it causes compression of the nerve fibers of the arm, leading to numbness, tingling sensation and paralysis in the night or the morning hours. So here is a table for your reference. The first column indicates the susceptibility to various factors like pressure, hypoxia and local anesthetics, and type of fibers most susceptible to that particular modality are A, B and C. So A fibers are susceptible maximally to pressure, B for hypoxia and C for local anesthetics. Next question on the same facts. Which of the following nerve fibers is affected by local anesthetics first? Let us see the options. A, parasympathetic, B, A, C, B and D, C. So already we have seen this table. Local anesthetics, the fibers which first are affected are type C fibers. So correct answer is option D, C fibers. Again the MCQ on the same fact. Most susceptible fibers to hypoxia is, and I think everybody will know the answer immediately, it is type B fiber. Okay, coming to the next question. Sensory fiber with maximum conduction velocity are A, C fibers, B, A alpha fibers, C, A beta fibers and D, A gamma fibers. So as you can see the responses from my students, again there was a lot of confusion amongst the different options. Most of the students don't pay attention to this part of the syllabus. So please understand just one table related to Erlanger-Gasser classification of the nerve fibers and you can answer n number of MCQs based on this. So here is a table for your reference. You are interested in sensory fiber with maximum conduction velocity. So it is A alpha fibers with conduction velocity up to 120 meters per second. Another question on the same Erlanger-Gasser classification. Type B nerve fibers are seen in A, preganglionic autonomic, B, somatic motor, C, motor to muscle spindle and D, sympathetic postganglionic. Okay, correct option is option A, preganglionic autonomic fibers. So please remember, preganglionic autonomic fibers are thinly myelinated type B nerve fibers and these are responsible for formation of white ramus, which you have studied in autonomic nervous system. Somatic motor fibers are nothing but nerve supply to your skeletal muscles. So these are A alpha fibers. A alpha fibers also are proprioceptive fibers. The fastest conducting tract in the body is spinocerebellar, dorsal spinocerebellar tract, which carries proprioceptive impulses. So those are also A alpha fibers. Option C was motor to muscle spindle. Muscle spindle fibers, efferent to muscle spindle are gamma fibers, gamma 1 and gamma 2, gamma motor fibers. So they are A gamma type of fibers. And sympathetic postganglionic fibers are unmyelinated type C fibers, which form grey ramus. Okay, next simple MCQ. compound action potential is property of A. Single nerve fiber, B. Sensory nerves, C. Motor nerves, and D. Mixed nerves. Yes, the answer is correct. It is the property of mixed nerve. When a mixed nerve is stimulated with suprarachal stimulus, recording electrode records multi-peaked summated potentials from different nerve fibers within the nerve. The separation of peaks and their amplitudes depend on the composition of nerve and the distance between the stimulating and the recording electrodes. The potentials from high velocity fibers like A alpha reach the recording electrode earlier, while those from slow conducting fibers like C fibers are last to reach. So you get multi-peaked appearance. So it is a property of mixed nerves. So if you have seen, many questions are constructed on Erlanger-Gasser classification of the nerve fiber. Okay, that's all for this session. Thank you for watching and see you in the next video. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.